Okay, right. So, hi, thank you very much for having me on, and apologies for the technical problems, but hopefully you can hear me okay. So I want to talk today about RNA vaccines for COVID-19 and the work that we've been doing at Imperial to start to bring RNA technology to the UK and look at the possibility for developing this for vaccines, and also thinking about the other kinds of applications that might be possible with this very interesting technology. So thank you for having me. So I'm just going to start very briefly with thinking about COVID-19 vaccines and immunity to SARS-CoV-2 and thinking about the characteristics of the ideal vaccine. We've heard already from Pedro about the adenoviral vectored vaccines and the encouraging results we're starting to get from that work. And the challenge of SARS-CoV-2 and the correlates of protection that we might need to think about in terms of developing a vaccine. And I wanted to talk about the landscape of COVID-19 vaccine research and in particular the huge plurality in the vaccine race. I don't think in the last 100 years there's been anything quite like it. And then talk to you about molecular vaccines and the work that we've been doing at Imperial on self-amplifying RNA vaccine. So why do we need a vaccine to tackle COVID-19? Now, vaccines, as we know, are remarkable public health tools. That's why we're all here today and why we've been working so hard over the past months to develop vaccines against COVID. So thinking back, I find this slide really remarkable. So in January, there were just a handful of cases of COVID-19 being reported. And the question in the UK was it going to come here? What were we going to see? And of course, we now know the answer to that question, sadly, with so many tens of thousands of deaths in this country and so much comorbidity. And vaccinating the world is a huge ask. And I think across the scientific community, we can agree that vaccination programs in one country is not enough. We're going to need to immunize everybody. And we can think about this as a global immune response to a novel infection. And really, vaccines are our most powerful tool to deliver an immune response that favors the host. And that's really what we're going to need to do with our immunization program. And I would put to you that it's not just a question of rolling out the first set of vaccines, that actually what we're going to need is a long-term public health strategy that includes probably several different vaccines in order to first engage the immune system and then maintain immunity against the virus with this constantly changing landscape epidemiologically and potentially virologically in the future. So the ideal vaccine would be safe and would be effective. And those are the most important parameters of vaccine. And then we can also think about all of these other things that we might want to ask of it. So we would want it to be well tolerated, immunogenic across all age groups and ethnicities, easy and very cheap to manufacture, rapidly and massively scalable, and give us lifelong immunity. I think it's very unlikely that any vaccine is going to give us all of those, but it may start to give us some of them. And just to get you to think about this in a bit more detail, when we're talking about efficacy, what do we mean? Do we mean efficacy against disease? Do we mean efficacy against infection? Do we mean efficacy against transmission? And as these vaccines start to get rolled out, that conversation will become increasingly important as the public health landscape changes. So there's still much to learn about immunity to SARS-CoV-2. And I'm going to give you an example here of why that is so important and why the landscape is complex and why we still need to continue research into natural infection. So influenza is very familiar to us, a respiratory virus which is endemic in the community and occasionally becomes pandemic. But there has been so much research on influenza that we do fully understand about the correlates of protection that are required and compared with, for example, something like HIV or hepatitis C or now SARS-CoV-2. where the correlates of protection are not fully understood, or if they are incredibly difficult to mimic. With flu, we can do this successfully. Correlates of protection in terms of hemagglutination inhibition titers, they're not perfect, it's a relative correlate of protection, but that allows us to relatively quickly develop vaccines against flu. We do not have that yet for SARS-CoV-2, which makes things much more difficult. But the last respiratory pandemic that affected the world was in March 2009. The first cases were reported in California and Texas. By September, there were mass immunization programs. So really the development of these vaccines, which were using traditional technology against flu, was very rapid because of this understanding that we have about what the bar is that we need to meet and to develop a vaccine, given all the caveats of that bar and in terms of it being a relative correlate of protection. And I've just put in this paper here where in China they vaccinated 100,000 people in 5 days. So SARS-CoV-2 is not the same thing, and it's not influenza A. And in order to develop a vaccine against it, we need to look at what we already know about coronavirus vaccines, and then look at what we can do in order to develop a pandemic vaccine. So there are several licensed coronavirus vaccines, but they are all for animals, and they all have their problems. As a general rule, coronaviruses in animals are diseases of the newborn or very young animals. So most of these vaccines are licensed for the newborn or the mother. And just here I want to point out the feline coronavirus, which Nana Marie is going to talk about a bit more later, in terms of why there was some concern about antibody-dependent enhancement and vaccine-dependent enhancement. So the feline coronavirus is actually an infection which is defined by a change in viral tropism that then causes an antibody-dependent immune complex enhancement of disease. And thankfully, we do not seem to be seeing this with SARS-CoV-2. It's not been described in the same way, but it does mean that for the feline coronavirus, a traditional vaccine was not going to be possible. And here are some other veterinary vaccines which have also been licensed. But I would argue that this knowledge hasn't really given us a huge amount of understanding about how to develop a vaccine against a human coronavirus, which is really quite different. So we can ask some questions about what protective immunity there is. There's a growing consensus that neutralizing antibody against the spike protein is what's required in order to prevent viral entry and therefore possibly even infection and maybe disease. And as more results come from our efficacy studies, we'll be able to test that hypothesis. But it's certainly the gamble that the entire vaccinology community has placed on developing a vaccine. And it is the reason why the vast majority of vaccines use the spike protein in either its pre-fusion conformation or unstabilized conformation in order to induce antibody. What do we know about the longevity of antibody responses and the likelihood that these vaccines will be protective in the long term? Well, there's conflicting data in natural infection, and we need to continually monitor this. However, we know from SARS and MERS that you do get clinically significant waning of antibody after 2 to 3 years. And so far, there have not been many cases of reinfection with SARS-CoV-2, but this is certainly something that we need to watch, and we need to understand when that happens, if there is any a change in the clinical appearance of the disease. So the landscape of COVID-19 vaccine research, traditional vaccinology is very much challenged. So here I'm showing growing viruses in eggs. That has particular problems. It's slow, it's expensive, and it has some problems with egg adaptation. However, as I showed earlier, we're very familiar with this approach, and we can utilize it quickly when we know what we're doing, for example, with the flu vaccine. But for SARS-CoV-2, it's a new infection, and that has led to huge plurality in the vaccine space, and essentially almost every platform that is available has been utilized in order to develop so many vaccines. This is a handful of them that are reaching clinical testing. There are many, many more that are still in preclinical testing. And both inactivated and protein subunit vaccines are being tested, including Novavax, where there's a large clinical trial ongoing in the UK. And then these more novel platforms, which include both RNA and DNA vaccines. And Imperial College is developing a self-amplifying RNA vaccine, and I'm going to talk a little bit more about that as part of the landscape in general of all RNA vaccines. So we want to think about working towards precision immunization. So where did molecular vaccines come from, and why has it taken up to this point for them to be a part of the landscape of vaccinology? So in the 1990s, they were first tested in mice and showed both protein expression and also that there could be some protection from infection. However, when dealing particularly with RNA, we need to be able to stabilize the molecule so that it can be introduced into the host. And that required some complex biochemistry, which work was done largely by Andy Gale and colleagues at Novartis. So how do they work? So a deoxyribose nucleic acid DNA vaccine or a ribonucleic acid RNA vaccine have both been developed. The principal challenge with DNA vaccines is that they have to get into the nucleus, and that is something that is very difficult to do in humans, and it is largely why, to some extent, DNA vaccines have not shown the initial promise that they had in animals. RNA vaccines, however, seem to be giving us a different readout, which is very encouraging. So the advantages of molecular vaccines is that they are readily modifiable. So you can easily swap out in and out an antigen in order to make it a new vaccine against possibly a new strain of a particular infection or a new infection altogether. And as we develop manufacturing capability, the cost and the scalability will start to be there in order to deliver vaccines to the whole world. And in terms of protein expression, the RNA vaccines allow very much a specific design, allowing you to express the protein in the conformation that you want it to be expressed in. And this is extraordinarily powerful, and I think is something that we should certainly be exploiting in the UK to develop not only vaccines, but molecular medicines as well. So what's a self-amplifying RNA vaccine? So a self-amplifying RNA vaccine you can see is a long chain molecule here of around 9,500 nucleotides. And because it has this self-amplification capacity, you get more copies of the RNA in the cell. And I've got an infographic that's going to show exactly what I mean by that. The plasmid DNA, as I said earlier, requires penetration into the nucleus for expression. And mRNA vaccines, which I will also talk about, require a higher dose because they don't self-amplify. So what has been chosen as the antigenic target for the vast majority of these RNA vaccines, in fact, all of the RNA vaccines that I'm going to talk about today, apart from one which has just looked at the receptor binding domain, the rest have all stabilized the spike protein in the pre-fusion conformation. And the reason for that was largely due to neutralization titers likely to be more effective at preventing viral entry, and also having the spike protein stabilized in its pre-fusion conformation would mean that you're less likely to develop antibody with FC activity. So the spike protein itself is shown here in this very nice publication from Daniel Rapp in Science. So I think what we've seen over the past 9 months is really a revolution in RNA technology. And there were already 2 companies that were quite well developed, and we've heard a lot more about them in the news. But there are several RNA vaccines that have been developed against COVID-19, and they all use a very similar approach. So here we've got the Imperial College vaccine, which uses a self-amplifying RNA in a lipid nanoparticle. I'll explain more about the importance of those lipid nanoparticles and what they might mean for vaccination as we go through the talk. And there's also several other companies. So CureVac, which is developing which has a non-modified mRNA. And then Pfizer-BioNTech, which you will be familiar with. They looked at two different vaccines, one that was against the RBD and the receptor binding domain just at the spike, and one which was against the spike protein in its pre-fusion conformation, similar to the Imperial College vaccine. And then the Moderna vaccine, also similar. There's one other self-amplifying RNA vaccine which is in development, which has got this interesting self-transcribing and replicating mRNA STAR technology, which is clearly patented. And then in China also they're developing an mRNA vaccine. And I just wanted to point out something here which I think is important. So here you can see that some of the companies are using a nucleoside modified mRNA, and the reason for that is to reduce innate immune sensing and therefore reactogenicity, and therefore increase the payload of the vaccine when it gets into its cell. That is something which I think we need to study and look at more in terms of understanding how these vaccines are working when they get into the cell. The Pfizer-BioNTech papers reported a difference in the reactogenicity between their two vaccines, which I think is very interesting, because the only difference is actually the sequence that they used, whether it was a receptor binding sequence or the full spike protein. And in the end, they went with the second of their candidates, and the BNT162b2, which is against the spike protein, largely because it was less reactogenic. So what do we know about these vaccines in preclinical data? Well, I think in a nutshell, they both induce neutralizing antibody, they both induce CD4 T cells and CD8 T cells, and they both at therapeutic doses induce protection in the lungs, and importantly, the Moderna vaccine also in the nose. And I think if that does translate into human efficacy, then that would be extremely important, because we would want to understand that better to know about not only infection but also transmission. And if we're going to have a vision of ending this pandemic, then we really need to be focusing on transmission in the long term, although the first goal is to end COVID-19 disease. And we've had some readouts about efficacy for both of these vaccines. The Pfizer-BioNTech vaccine has met its primary efficacy endpoint, and showing a protection from disease, and the Moderna vaccine is not far behind. And in terms of tolerability, these vaccines do seem to be well tolerated, although the Moderna vaccine has shown increased reactogenicity with the second dose. And I think, again, that is something that we need to study. And this is why I am keen that when we start to roll these vaccines out, that we do that as part of close monitoring, perhaps even of a phase 4 trial, so that we can understand these novel platforms better. I think across all the studies that have reported, and also Sputnik 5, I should mention here as well, is that what we want to understand initially is, are we seeing severe cases of COVID-19? And as a general rule, apart from one in the Pfizer-BioNTech group, that we've not seen severe cases in the vaccinated groups. And that is what we need to look for very carefully when we start to roll out this first wave of vaccines, the first hurdle being to reduce COVID-19 disease in the community. So the Imperial saRNA program is using SARS-CoV-2 as a test case in order to develop this platform and to look at whether we can achieve a vaccine that can be manufactured at low cost and at scale and using a low dose. So the self-amplifying component of the vaccine is alpha virus derived. There are no structural proteins. And as I said earlier, it contains a transgene for the antigen that can easily be swapped in and out. So the timeline of making the vaccine has been very fast. And when you consider that we had not done this before at all in humans, it had only been developed in animals previously. And so we were really working against a backdrop of limited clinical experience globally with these RNA vaccines. There'd only been a couple of clinical trials of mRNA, and no experience of self-amplifying RNA. So that's really what we've been able to achieve in the last 6 months. And I'm going to show you a little bit more about how the vaccine is designed. So here you can see the 4 non-structural proteins of the amplicon, and that is what amplifies the vaccine when it's expressed in the cell. And there, the transgene, in this case, it's just luciferase, but we could put in anything we wanted there. And so I'm going to explain a little bit more about how this works. So in our case, the vaccine antigen is the spike protein, which is stabilized in its pre-fusion conformation. Here is a cell, and I will show you how the vaccine enters the cell. So it uses a lipid nanoparticle with the RNA, this self-amplifying RNA. Remember, I said it was a long chain molecule inside the lipid nanoparticle, which then fuses with the membrane and enters the cell. The payload of the vaccine is released and then encodes the 4 non-structural proteins, which makes this amplicon, which allows the positive and negative strands of the RNA to be transcribed within the cell. And that makes many more copies of the transgene, which are preferentially transcribed. And then expression of the spike protein, as you can see here. And what that's allowed us to do in animals is to test this vaccine in a mouse experiment, which was published in Nature Communications. And here you can see the dose response. So, and in fact, it's interesting with this study. So even with the DNA and electroporation on the left here, we were getting quite good responses. You can see with the rabies control, there was no SARS-CoV-2 specific IgG, and there's a very nice dose response, which is across the different doses here, and and higher than here you can see convalescent serum. And when we look at the neutralization titers, those are also very similar in the mouse experiment, going from 10 micrograms here to 0.01 micrograms here. And here, patients who've had SARS-CoV-2 and who have convalesced. And on the right here, I'm showing that there is a correlation between the neutralization and the binding titers, which allows us to infer a little bit more about the functionality of binding antibody. And what I want to point out here is, why are we trying to develop this self-amplifying RNA? Well, it's really in this in these doses here. This is much less than the Moderna vaccine, and that is why we thought that it would be important to develop this, that we could give a lower dose. So here, 10 micrograms, going all the way down to 0.01 micrograms, and still in the mouse experiments getting an antibody response. So clinical trials. So as I said, designing this clinical trial was a real challenge, because we were starting literally from scratch. We had no clinical data.
So, Kat, I'm really sorry to interrupt you, but we're running out of time.
But we had all
and we need to move on to the next session.
And also the
so, can I ask you to draw your presentation to a close?
So that has meant that we have designed the trial with caution and also with scientific rigor. And the phase 1/2A clinical trial is looking at safety, immunogenicity, and dose finding. And it's quite a large trial. We've enrolled volunteers aged 18 to 75 years, and we're fully recruited now to this part of the protocol. And we are very grateful to our other trial sites who've supported us in this work. These are the groups that we've studied. So we've done a first dose escalation with a prime boost at 0.1, 0.3, and 1 micrograms, and then a second dose escalation once we knew that these lower doses were well tolerated and were safe at 2.5, 5, and 10 micrograms. And that dose escalation takes time. So essentially, you enroll a sentinel participant, and then phone them at 48 hours, and if they're okay, you then vaccinate another 3 people at the same dose level. And we've done that both for the prime and the boost, and we've done that for all these different dose levels. So really being very rigorous in terms of how we've evaluated the dosing of this vaccine. And I think that that is going to provide a very useful data set for developing other mRNA and self-amplifying RNA vaccines in the future. And it has been a strength of this trial, but it does take time to do that. Having done that, we were then able to expand the number of people that we've tested these doses on. So looking at the cohort B here, where we've got a larger numbers of people, where we've tested the 6 different dose levels across people, healthy volunteers aged 18 to 45. And then we have the phase 2 component of our study, which is an expanded safety evaluation, where our participants got 1 microgram after we completed part A of the study, and now they have had 10 micrograms after completing part B of the study, to really test whether we see this vaccine is safe and tolerable at both these dose levels, and also to look at the immune responses. So this is the trial design. The large majority of the safety and dose escalation work has been done at the Imperial CRF, where we do translational work for vaccines, as Rajeka said earlier. This has been completed now, this part of it. So the screening, the enrollment, the dosing, and the safety, tolerability, and immunogenicity assessments are all now forming part of our data set. And then we expanded to multiple centres, as I said, where they gave the 1 microgram prime dose. And then moving forward to the next part of the study, where we used the higher dose levels from 2.5 to 10 in a similar dose escalation, dose evaluation study, and established the highest tolerated dose, which was 10 micrograms, and then offered that to our expanded safety cohort. Our safety and immunogenicity readout is expected later in this year. So far, it's been well tolerated, and we are seeing more reactogenicity at the higher dose levels, which is what you would expect, and is similar to what was seen with the other RNA vaccines. That is where we are at the moment. I'm looking forward very much to sharing the data with you once we have a full data set. And as you can see, because of where we were in terms of clinical experience, that has meant that we have taken this approach that is both robust and cautious, and will give us a very detailed data set to understand how RNA vaccines behave in human beings, to allow us to develop not only a COVID vaccine, but also the technology in the platform. And I just want to leave you with a different thought, something more to do with vaccine rollout. And because I think this is a conversation that we should be having within the CVN UK, thinking about adult vaccination, and with the JCVI and the other stakeholders in terms of mass immunisation. So COVID is characterised by these 4 different levels of severe disease, mild disease, asymptomatic infection, and no infection. And a vaccine will move us across these different caseloads. And really, when we see a severe disease in ICU, that's just the tip of the iceberg. We know that that means that there is a significant amount of transmission happening in the community. So the ideal vaccine would completely prevent infection. And that would mean that we would move the population towards no infection and towards eradication completely of this infection, and also therefore disease. But however, it's much more likely that initially we'll be going to be getting disease modifying vaccines, as I showed earlier, and that will move the needle down from severe disease through mild disease towards asymptomatic infection. But what it will not do is prevent transmission. And so just to make one more point about that, if we do that, it's quite possible that because people who were previously symptomatic then have mild disease or asymptomatic, may be more likely to transmit. And that is why we must have very robust immunosurveillance and virological surveillance when we roll out these vaccines. So this has been a trial very much conducted in the public eye. We've done masses of outreach, masses of education, and huge amounts of media, but by far the most fun was Operation Ouch. For any of you who have children, you'll know what I'm talking about. For those of you who don't, these are a pair of infectious disease doctors at UCL who run a children's programme on the BBC, and our programme that we've done with them will be coming out next year. And thank you very much.
Thank you very much, Katrina. That was an excellent talk.